My name is Andrew, and I will be talking about Valonia today. So, for the basic taxonomy of Valonia, it is a eukaryote and a plantae as well. Its phylum is Chlorophyta, and class is also Lupophyceae. Its order is Cladophorales, and its family is Valoniaceae, but it was previously under Cyclonoclades. So, its genus, or not, its species number, though, according to AlgaeBase, there are only about 50 species of Valonia that are characterized. There used to be more, but however, genetic testing has proven that some of those species are not actually Valonia. So, like other Cladophorales, it has sporic meiosis and is characterized by its alternation of generations between the diploid zygotic phase and the haploid sporic phase. And I did not have a good picture of a Valonia itself's undergoing the life stages. However, this is a pretty accurate representation of the general formation of Valonia. Its other general characteristics, though, is that it is multinucleate as well as multicellular. And it undergoes exogenous cell division, where they're characterized by the cells dividing inside of its parent cells, while they also stick out of their former parent cells as well. However, an interesting thing that they do is cytoplasmic streaming, where the cytoplasm inside of the algae actually stream all throughout the algal body. So what that does is that it speeds along organelles and other proteins and materials throughout the cell and throughout the entire body, wherever it is needed. And additionally to that, they possess rhizoids, which they use for attachment at its base. So Valonia does not really have that many practical applications, or at least I haven't found any much research regarding them specifically in terms of practical applications. However, one interesting study that I found was in terms of their rhizoid development. So what they found was that they could artificially induce rhizoid development inside any part of the Valonia. As long as they had contact with the substrate, the rhizoids would differentiate from those cells, and they would attach to those substrates. And this, they found that in a study by Elvira et al. In 2012, they found that the soft substrates and dark incubation seemed to promote rhizoid development the best, so they weren't just growing inside light environments. However, when they were exposed to light, cytoplasmic streaming would be induced inside those rhizoids and actually force chloroplasts into them so that they can photosynthesize through those rhizoids. They also did some electrical stimulation tests. This was a different group by Wang et al. They did this in 1993 as well as in 1995. So what they did was they would subject Valonia to electrical stimulation in order to map out specific ion channels. So what they did was they attached an oscillating electrical current to the Valonia cells, and they mapped out how the ions would be moving throughout the body by observing potassium and chloride movement in the potassium, and they determined that the electrogenic transport can be driven by artificial currents, not just through natural metabolic processes. Furthermore, they also found how they inhibit turgor pressure to make sure that they have a constant pressure and are not overdoing it, and they're not swelling too much, or they're not being deflated by the constant changes in salinity that they experience in waves. So what they did was they would shock the Valonia and cause them to stimulate different levels of salinity, and they would experience changes in turgor pressure as well as changes in overall body morphology, which was an interesting finding that was corroborated with another study by Binder et al. In 2006. So they found that the alternation of electrical currents can cause alternation of ion channel flow. So they demonstrated that Valonia were able to alter turgor pressure based on induced electrical stimuli, and they found that they could also induce that and cause that based off of different metabolic processes that occur throughout the body while it is living. So that is pretty much all that I have for Valonia. I thank you for your time. Here's my literature cited. Any questions?